Hey, hello guys, welcome to IQCIVILS IAS Academy, a great coach transforms life. Today, we are going to discuss a very interesting and unique and important topic from UPSC Science and Technology syllabus. The name of the topic is Three Parents Baby. The name itself is unique. Normally, we know that there are two parents for a child or for a baby, but here the child has three parents. So what exactly this mystery is, because science is all about mystery and science is also about resolving the mysteries which are not solvable by the common sense of the humanity. So this very interesting topic of three parents baby will be covered by me, Dr. Sachin Kadam, faculty at IQCIVILS IAS Academy, Guwahati. This is our elite panel of faculties from Delhi that includes Sunil sir, Sachin sir, Anuj sir, Alok sir, Yogesh sir, Rizwan sir, Vikram sir and Nitin Kumar sir. We are starting our new batches from 25th May and 5th of June 2023. These batches are available offline as well as online, and you need to register yourself to book your seat in these batches. Okay, for that you need to call on this number. This is our director Mr. Anuj Singh sir. This is our admissions manager Lisa Tamuli, and this is our front office holder Bikas Deka. Like this video, share this video and subscribe to our YouTube channel for more and more interesting videos on our YouTube channel. Admissions are in process for April-May batches for UPSC and State PSC 2024 examinations. IQ Civils IAS Academy is running many government projects since 2018 in collaboration with Assam Administrative Staff College so that meritorious students from Assam can easily crack UPSC as well as other state services examination. This is the Google review given by the students to us, 4.9 star. That is rare and unique for UPSC CSC examination. So you can see the quality provided at IQ Civils IAS Academy is unmatchable. So why this three parents baby was in news? So recently a three parent baby was born in United Kingdom using in vitro fertilization. Now what exactly this three parent baby is? It is a technique to create and to have a child without passing on mitochondrial diseases caused by faulty mitochondria of the mother. So first point you need to remember, three parent baby is a type of gene therapy, okay, in which we replace defective mitochondria, okay, we replace defective mitochondria with healthy mitochondria. Clear? So defective mitochondria is replaced by healthy mitochondria, and that type of gene therapy is known as three parent baby, or it is also known as mitochondrial gene therapy, MGT, or mitochondrial replacement therapy, MRT. So to treat mitochondrial genetic disorders, this three parents baby technique is used. The mitochondrial diseases are long-term, genetic, often inherited disorders that occur when mitochondria fail to produce enough energy for the body to function properly. I will explain you what exactly mitochondria is. What is mitochondria? It is the cellular structure that provides energy to cells. So first of all, this is eukaryotic cell, the type of cell that is present in your and my body, or type of cell that is present in plants, that is present in every animal, eukaryotic cell. In an eukaryotic cell, there is a nucleus, and inside this nucleus, DNA is present. Along with nucleus, there are different cell organelles that are present in eukaryotic cells, such as endoplasmic reticulum. It is involved in secretion of proteins. Mitochondria, it is involved in energy production in eukaryotic cell. That's why mitochondria is known as powerhouse of cell. Okay, then lysosome, this is mainly involved in the destruction, or you can say breakdown of the waste generated in the eukaryotic cell. Sometimes this lysosome is capable to digest whole dead eukaryotic cell. That's why we call this lysosome suicide bag of the cell. What we call this? Suicide bag of the cell. Okay. Yes. Then this is Golgi apparatus. It is involved in the packaging of the protein. And there are many other cell organelles. So see, even this ribosome is there, which is very important. It is involved in the protein synthesis. So what happens in a eukaryotic cell? Ribosome will synthesize protein. That protein will be secreted by endoplasmic reticulum. That protein will go to the Golgi apparatus, and packaging of that protein will take place in this Golgi apparatus. And during this protein synthesis, protein secretion and packaging of protein, some waste will be generated, and that waste will be managed by lysosome. But for all these processes you need energy, and that energy is provided or generated in mitochondria. And if you observe this mitochondria, here you can see mitochondria has its own ribosome, mitochondria has its own DNA, and mitochondria has its own membrane, outer membrane as well as inner membrane. So mitochondria in itself is a type of prokaryotic cell which has its own DNA. So if there is any defective gene on this mitochondrial DNA, so this mitochondria will not be able to work properly, and if this mitochondria is not working properly, then this mitochondria will not be able to generate proper energy, so the functioning of whole cell will be affected. That's why if someone have defective mitochondria, if someone have mitochondrial genetic disorder, so we need to replace the defective mitochondria with the healthy one, then only that person can survive. And that's what we do in three parents baby. So what we do, three parent baby, this is mother's egg, okay, and this mother's egg has defective mitochondria. This red color means this has defective mitochondria. This is donor's egg, and inside this donor's egg mitochondria are healthy. So what we do, we remove nucleus from the donor's egg. So now we have a donor's egg with healthy mitochondria but without any nucleus or without genetic DNA which is present in the nucleus. Then we remove the nucleus from the mother's egg and insert this mother's nucleus into the donor's egg. Now we have an egg that has DNA of mother and mitochondria of donor, and mitochondria has its own DNA. So DNA present in the mitochondria belongs to donor, DNA present in the nucleus belongs to mother, and now by using father's sperm we will fertilize this egg. So here you got nucleus that contains DNA of mother and father, plus you have mitochondria that contain DNA of donor. So that's why this fertilized egg, what we call this zygote, contains the DNA of three people, mother, father and donor. That's why we call this three parent baby. In this case, we created three parent baby before fertilization. How? Because we transferred the nucleus of the mother's egg before fertilizing it into the cell of the donor's egg. That's why we call this maternal spindle transfer. In pronuclear transfer, what we do? We fertilize the egg of the donor with father's sperm. We fertilize the egg of the mother with father's sperm. Now we have egg with mother's and father's DNA but defective mitochondria. We have another egg with father's and donor's DNA but healthy mitochondria. So we remove this DNA of father and donor and we add the DNA from mother and father that is present in this egg into this egg. Now we have an egg that has mother's DNA, father's DNA in nucleus, and mitochondrial DNA from donor. That's why three parents baby. In this case, we transferred nucleus of the cell after fertilization. This is known as pronuclear transfer. In this case, we transfer DNA from the egg to another cell before fertilization. This is known as maternal spindle transfer. And this way, three parents baby is created. Okay. So one by one, what is three parents baby? This technique is a mitochondrial replacement therapy or mitochondrial gene therapy in which we replace the defective mitochondria present in mother's egg with the healthy mitochondria from the donor's egg. That can be done before fertilization or after fertilization. What is the main role or main function of mitochondria? Mitochondria is mainly involved in the production of the energy. Clear? Now I know it is a bit difficult to understand. Now I will explain it in a different way. See, this is donor's egg with nucleus and healthy mitochondria. This is mother's egg with nucleus and defective mitochondria. In maternal spindle transfer, what we do? We remove nucleus from donor's egg. So we have donor's egg without nucleus but healthy mitochondria. Then we remove this nucleus from mother's egg and transfer this nucleus to the donor's egg. So now this egg has nucleus from mother or nucleus DNA of mother and healthy mitochondria from donor of donor. We call this mitochondrial DNA. Okay. Now, if you fertilize this egg which contains DNA from mother in the nucleus, DNA healthy mitochondria from the donors with the sperm of the father, so you will get a zygote that has DNA from mother and father, nucleus that has DNA from mother and father, and mitochondria that has DNA from donor. So this is the three parent baby. This technique that I just explained you, this is known as maternal spindle transfer because in this case the DNA or nucleus that we transferred was only from mother. You can see here we transferred DNA of the mother into the donor's egg, and this is done before fertilization. Okay, this is known as maternal spindle transfer. Then there is one more technique. I will explain you that technique also to you. See, this is donor's egg with the nucleus and healthy mitochondria. This is mother's egg with the nucleus and defective mitochondria. This is defective mitochondria. Now what we will do? We will fertilize, okay, we will fertilize both the eggs with the father's sperm. So you will get two zygote. One zygote that has nuclear DNA from half from the donor and half from the father and healthy mitochondria. You will get another zygote that has DNA in the nucleus, half from the mother and half from the father. But the problem with this zygote is mitochondria in this zygote is defective. Okay, now what we do? We remove this nucleus. Okay, we removed this nucleus, so we got a zygote cell with healthy mitochondria and no nucleus. Now you will insert this nucleus of mother and father into this egg. So you will have a nucleus that has DNA from father and mother and mitochondrial DNA from donor. So this is a 3-parent baby because this baby has the DNA from 3 person. And this, in this case, transfer is done after fertilization, and this transfer is known as pronuclear transfer. What we call this? This transfer is known as pronuclear transfer. Clear? So mitochondrial gene therapy or 3-parent baby, this test or this experiment can be done before fertilization or it can be done after fertilization. Now I will explain you the mechanism. What is the mechanism? Researchers do this by exchanging the deceased mitochondria of a prospective mother with those of a healthy unrelated donor, the third parent. The procedure replaces a small amount of faulty DNA in a mother's egg with healthy DNA from a second woman, so that the baby would inherit genes from 2 mothers and 1 father. And these, in this case, 2 mothers, nuclear DNA belongs to the mother and mitochondrial DNA belongs to the donor. The idea is to prevent certain genetic diseases from being passed on to children. The technique used is called maternal spindle transfer, or it can also be called pronuclear transfer. Maternal spindle transfer, this term is used if you are doing this before fertilization, and pronuclear transfer, this term is used if you are doing this after fertilization. Okay, so this, the technique is called maternal spindle transfer, in which maternal DNA is put into the egg of a donor woman, which is then fertilized using the father's sperm. The procedure was the procedure was developed to help existing in vitro fertilization treatments in which mothers have mitochondrial diseases. And remember, all the mitochondria that your or my cell have, all the mitochondria present in our cell, they are completely inherited from mother. So if some female has mitochondrial DNA diseases or disorders that are related to mitochondrial DNA, then the every child of that female, male or female child, every girl or boy child of that female will have mitochondrial disorder. Okay, what is in vitro fertilization? It is a medical procedure in which mature egg cells are removed from a woman, fertilized with male sperm outside the body, and inserted into the uterus of the same or another woman for normal gestation. Okay, if you are inserting it into the uterus of another woman, that will be known as surrogacy. And if you are inserting or implanting that baby in the uterus of mother only, then that is simple IVF. Okay, Britain became the first country to allow for a 3-parent baby in 2017, and the first 3-parent baby was born in 2023. The case related to 3-parent baby was also fought in Mexico, Central American country. There also the issue of this 3-parent baby arose, and in UK also, but UK court legally allowed three parent baby. Why they legally allowed three parent baby? According to UK court, if there is a treatment available for the genetic disorder, it should be made available. Now, who was opposing three parent baby? Obviously, religious leader. Church was opposing three parent baby. Why they were opposing three parent baby? Because according to them, a baby can have only two parents. How you can, how you are creating a baby with three parent? So भाई साहब, it is science, तो कर रहे हैं, अब क्या करें? अब देखिए, even if this is a three parent baby, the genetic makeup of that baby will be completely of mother and father, because DNA present in the nucleus belongs to mother and father. Only mitochondrial DNA belongs to donor, and this mitochondrial DNA, it has no physical character. There is no physical changes in physical appearance of the baby. Okay, so this religious leader or people who follow religion, they are illogical. They always have problem with the science, because science उनकी दुकान बंद कर देता है। रिलीजन बेस्ड ही किस चीज पे होता है? फियर, डर। ऐसा करोगे तो ऐसा होगा, ऐसा नहीं करोगे तो ऐसा होगा, मतलब डरा कर रखता है रिलीजन। और साइंस कहता है यू नीड नॉट टू फियर, देयर इज नो नीड टू फियर, जस्ट फॉलो योर रीजनिंग, just follow the science and you can have a really good life. Okay, religion maximum can help us in our mental issues. If you are unable to deal mental issues by science, so you can take help from the religion. Otherwise, I personally feel religion is more like a individual's connection to the unknown power. There is no need of middlemen, that is pandit, molvis, or jathedars, or padris. No one is needed. You can directly connect to your God. Okay, these middlemen are not needed. So there were few questions which were related to the genetics or which were related to the genome. So the first question is, this is a question of biotechnology. In the context of the developments in bioinformatics, the term transcriptome, sometimes seen in the news, it refers to. So see, this is DNA. Information on DNA is first copied on mRNA. This is known as transcription. Then from mRNA, this information get translated into protein. This is known as translation. And if there are 30,000 genes, so there will be 30 different types of mRNA, 30,000 different types of mRNA. So mRNA is the product of transcription. That's why all the mRNA present in human body are known as transcriptome. Okay, so that's why, just a minute, I need to take this call. So see, in the context of the developments in bioinformatics, the term transcriptome, sometimes seen in the news, refers to the full range of mRNA molecules expressed by an organism. Answer is B. We are going to the question number 2. Again, a question related to the genetic engineering. The Genetic Engineering Appraisal Committee is constituted under which act this GEAC is constituted or GEAC is created. Okay, it is actually created under Environment Protection Act or EPA 1986. Environment Protection Act of 1986. Under this act, this GEAC is created. That's why your answer is C. And now this question is directly from the MRT, mitochondrial replacement therapy, and this question was asked in I think it is in 2019 or 20, most probably. I don't remember exactly, but yes, please read this question. In the context of hereditary diseases, consider the following statements. Number 1, passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of egg. Yes, the statement is correct. Why the statement is correct? Because If you are doing this before fertilization, we called that maternal spindle transfer. If you are doing this after fertilization, we call that pronuclear transfer. So yes, both are correct. It can be done before fertilization as well as it can be done after fertilization. First statement is correct. Second, a child inherits mitochondrial diseases entirely from mother and not from father. Yes, if all the mitochondria present in our body are inherited from mother, then obviously mitochondrial diseases will also be inherited from mother only. So second statement is also correct. So which of the statement given above is are correct? Obviously your answer should be C. Both 1 and 2, these statements are correct. Okay. So this is about three parents baby, guys. Three parents baby is a very important topic. It was in current affairs, so I explained you properly. In crux, three parent baby mitochondrial replacement therapy, mitochondrial gene therapy is used to treat the genetic disorders that are linked to the DNA present inside the mitochondria, which is called as powerhouse of the cell, which is responsible for generation of energy in eukaryotic cells. Okay. Notes of this video are available on this Telegram channel. To subscribe, so subscribe to this Telegram channel, like this video, share this video, and subscribe to our YouTube channel of IQ Civils IAS Academy. We are starting our new batches from the 15th of May and from 6th June. So if you want to book your seat in any of these batches of the May batch or of the June batch, then what you need to do, you need to call on this number, either this number or on this number, and book your seat. Once you book your seat by calling on this number, you can access the best quality UPSC teaching or coaching on IQ Civils IAS Academy, online as well as offline. So that's it from my side for the today, guys. If you have any doubts, you can ask your doubt in the comment, because whenever this video premieres, I watch that video, and if you'd ask any doubt, I do answer those doubts in the live chat or those doubts in the comment box. Okay, so that's it from my side, guys. Thank you very much. uh, Study hard, work hard, and ensure that your hard work and your study and our teaching gives result either in UPSC or in APSC examination. So yes, like this video, share this video, and subscribe to our YouTube channel without missing it. Okay, so that's it from my side, guys. Bye bye, take care, sayonara, see you.